Hi everyone, and you're very welcome to today's Saturday session. Our topic for today is going to be DNA structure, replication and profiling, and then we're going to look at protein synthesis as well, all part of Leave Insert Biology. Now, before we get into today's content, the idea of DNA structure, DNA replication, DNA profiling, these are very, very common exam questions which have come up over the past couple of years. So as we go through the content today, we're going to look at some material. We're then going to look at some exam questions based on the stuff that we've looked at. And then at the end, I'll show you where you can access more content just like what we're going through today. So to start off with, for the structure of DNA, one really important thing to know is that DNA stands for deoxyribonucleic acid. Where this comes from, the deoxyribo part, is the sugar that's present in DNA, which is deoxyribose. Okay, that's one thing you are expected to know, and it is actually part of an exam question which comes up as well, to know a difference between the structure of DNA and RNA, and the sugar is actually one of the differences there. So DNA stands for deoxyribonucleic acid. It always takes the form of a double helix, so if you picture a twisted ladder like we have over here on the right hand side. And just one thing I want you to take note of at this stage, and we will come back to this in a couple of minutes, is if you look towards the center, these here are the base pairs. And if you look the whole way down, it's color coded. It's not going to be these actual colors inside our bodies, but they are color coded the whole way down. So you can see here the orange is always paired off with the green, and this purple color is always paired off with this blue color over here. Okay, so keep that in mind because that's going to be really useful for us and really important in a couple of minutes' time. Now, how do we actually form this double helix, this twisted ladder shape that we can see? Well, the sugar that we were talking about just a second ago, the deoxyribose, that actually forms part of the backbone which we have out here. So we have one part there and we have another part running up here. So that backbone there is made of the sugar, and it's also made of a phosphate. Okay, both of those, the sugar and the phosphate, are constituents of the basic unit of DNA, which is called the nucleotide. And we can see that down here in this diagram here. So this double helix up here is made up of several of these nucleotides. This nucleotide is the building block, essentially. Like the amino acids are the building blocks of proteins, the nucleotides are the building blocks of this. So what does a nucleotide actually consist of? Well, we have our pentose sugar, a 5-carbon sugar, which is here. That's our deoxyribose. We have a phosphate group attached to it, and we also have a nitrogenous base. Okay, that nitrogenous base there can be one of 4 different things. The phosphate never changes, the pentose sugar, the deoxyribose, never changes. The nitrogenous base is the only thing that ever changes, and it's going to be one of these 4 things which we can see in here. Now, we mentioned about how we can have 4 different base pairs. Those 4 base pairs that we can have are adenine, guanine, thymine, and cytosine. Okay, and they're always going to pair off with one that's complementary to it. Okay, that's where the idea of complementary pairs are coming from. So we can see over in the diagram here, we can see that they're shaped a little bit differently. They're shaped complementary to each other, just to give us the idea that adenine always joins or pairs off with T, which is thymine. Okay, adenine and thymine always go together. Guanine and cytosine always go together, and you can see that here towards the center. They have approximately the same shape as each other, so they're a complementary fit. Now, how are we going to remember which one pairs off with which? Well, if you have a look at this diagram down here in the bottom right hand corner, any geography students might recognize it. That's the Giant's Causeway. So one simple way of remembering which pairs with which, if you just remember at the Giant's Causeway, and just look at the first 4 letters that we have there. We have A, T, G, and C. So adenine joins with thymine, guanine joins with cytosine. Now, collectively, these can be paired off into 2 different groups, and you are expected to know these as well. So adenine and guanine collectively are known as the purine bases, whereas thymine and cytosine, just remember they have the letter Y, they're collectively referred to as the pyrimidines. Okay, and you are expected to know those 2 terms as well. So adenine and guanine are the purines, thymine and cytosine are the pyrimidines. Now, how do we actually form this double helix structure? Well, it always takes the form of the double helix. So if you picture back to that twisted ladder, the base pairs are going to begin to join towards the center. And again, you can see that a little bit more clearly now in the double helix. Adenine joining to thymine, guanine joining to cytosine. They're complementary pairs, so they have a similar shape to each other. Now, in here towards the center, how they're actually held together, it's with hydrogen bonds. And again, that's a really important thing to know for an exam question. Commonly comes up when DNA is asked, is to name the type of bond or how are they held together. Hydrogen bonds being the answer. Now, who actually did the study behind all of this to come up with the double helix structure? Again, that can come up as part of an exam question. There was actually 3 scientists that worked in it. 2 of them were given credit at the time. That was Crick and Watson. But Rosalind Franklin actually had done a substantial amount of work as well at the same time, just wasn't given the credit for it at the time. So Franklin, Crick and Watson all worked on the idea of the double helix structure, where the outside strand, so this backbone, that's the sugar, the deoxyribose and the phosphate, and the insides then was the base pair. Now, this DNA here, we saw that a couple of minutes ago in the double helix structure, how all the base pairs are lined up here towards the centre. The sequence that those base pairs in is really important for us because that actually translates across to proteins, okay? So this is all going to come back to the idea of genes and inheritance. So chromosomes are what genes are found on. Where are these chromosomes found? Well, inside the cell, you have your nucleus. Inside your nucleus, you have your genetic material, and that's where your chromosomes are going to be found. Now, here's an example of chromosomes. On those chromosomes, we have these bands that appear across them. Those bands that you can see are genes, okay? Those genes, they're what control inheritance. They're units of inheritance, and they control different things, inheritable factors that can be passed off to you from your parents. So, for example, hair colour, eye colour, and so on. Now, there were about 20 different proteins, 20 specific proteins that can be encoded for, depending on the order of the base pairs within the DNA sequence. Each one of them has a specific function inside our bodies. Okay, so DNA codes for proteins using 3 base pairs at a time. So that can be referred to then as a triplet or a codon. Really, really important to know that there as well. But sometimes you could have a sequence of letters that just don't actually code for a protein. Okay, this is referred to as junk DNA. So again, that's actually a common exam question which has popped up before. What do we mean by junk DNA? It's just a sequence of DNA or DNA that doesn't encode for protein. Okay, now what do we mean by encoding for proteins? Well, if we have a look over here, this is how we actually translate across from our base pairs into our proteins. So if we were to have a look up here at our DNA sequence, let's say, for example, we had thymine here first of all, then we had cytosine, and then we had guanine. Okay, that's just an example. So we would have TCG. Well, down here we would look, the first base pair we had was T, the second one was C, and then we had G. So TCG is here. So that's actually serine. So TCG would code for serine. Now, if you have a look along here, you will see the word stop. If you were to encode for one of those, so for example, TAA, if you thymine, adenine, adenine, or thymine, adenine, guanine, that means it's actually going to stop coding for proteins. Now, actually opposite to that, we do actually have a start codon as well. One of the start codons we have would be ATG. So it's not actually labeled there, but if we look for ATG, we can see it's there. So if we do come across a stop codon, your body will actually completely stop coding for proteins, okay? It could go several base pairs along and it won't actually start coding for proteins again until it gets back to a start codon, okay? So anything that doesn't encode for protein then, again, we refer to that as junk DNA. Now we're going to have a look at the structure of RNA now as well, because we're going to need this when we look at protein synthesis in a little while. So RNA stands for ribonucleic acid. Okay, a little bit different because the sugar that RNA has, it's not deoxyribose, it's actually just ribose. Again, you could be asked as part of an exam question to give differences between DNA and RNA. One of those could be that the sugar in DNA is deoxyribose, the sugar in RNA is ribose. RNA has 3 of the same base pairs as DNA. It also has adenine, it also has guanine, and it also has cytosine. However, it does not have thymine. Thymine is replaced by uracil. Okay, that's a second difference between DNA and RNA. DNA has thymine, RNA has uracil. A third difference between DNA and RNA, we know that DNA is double-stranded. Picture the double helix that we seen a couple of minutes ago. RNA, however, exists just as a single strand. Okay, and we can see that in the diagram over here. It's just one strand that we have. RNA will actually also be complementary all the time to DNA. So the reason that this is an A over here is because there was the thymine over here on the DNA. The reason this is a G is because there was the C over here. Remember, U replaces T, so U is opposite an A, and C is opposite a G. So this is complementary to a strand of DNA, which we would have had over here. And you can actually see that down here, okay? You have DNA here, RNA over here. So in DNA you had G, that means we have a C. In DNA we have an A. If it was another DNA strand on the other side, we would have a T, but because it's RNA, we have to replace that with uracil, which is a U, and that continues the whole way down. Okay, and that's essentially what's being told down here. If you have an A, it's going to be a U in RNA. If you have a G, it's going to be paired off with a C, A paired off with a U, and so on. Okay, so a couple of differences there between DNA and RNA. DNA is double-stranded, RNA single-stranded. DNA has deoxyribose, RNA has ribose. And then 4 of the base pairs that we have in DNA, 3 of them are the same in RNA. We have A, we have G, we have C, but T is replaced by uracil. Now, as part of your study of Leaving Cert biology, you actually have to perform an experiment to isolate DNA from plant tissue. It can come up as an exam question quite often, but the questions on this are actually quite repetitive. And usually how these questions come up, and we will see this, is it's usually asking you for the why of every step. Okay, so if you have a look at this overall experiment setup, you would have done it very, very similar. Some steps might have been slightly different, but some of the plants that you would have used to isolate the DNA from, you could have used an onion, you could have used a banana, you could have used a kiwi, you could have used celery, something along those lines. You would have used washing up liquid, you needed table salt, you needed a protease enzyme, and you also needed ice cold ethanol as well. And each one of those pieces there, the washing up liquid, table salt, and so on, the question that usually comes up on this experiment is, why did you add washing up liquid? Why did you add table salt? Why did you add the enzyme? And why did you add the ethanol? And we're going to go through that now. So if you were to think back a couple of minutes ago to where the DNA actually exists inside the cell, it's located inside the nucleus. Okay, remember, we're dealing with a plant cell in this experiment. So not only do we have a cell membrane, we have a cell wall, we have a cell membrane, and we have a nucleus. Okay, we have to get through all of that before we can access the genetic material or the DNA. It's inside the nucleus. Okay, so we have to get through the cell wall, we have to get through the cell membrane, we have to get through the nucleus. How are we going to do that? Well, the first thing you're going to do is using a scalpel or using a knife, you chop whatever you're using, the onion, the kiwi, whatever it is, you're going to chop that into small pieces. Why are we doing this? We're breaking open the cell wall. Okay, that's the first line of defense we have to get through essentially. We have to break open that cell wall. So if you're asked why did you chop it, we broke the cell wall. Following on from that then, we're actually going to add in some washing up liquid and we're going to add in some salt. A couple of reasons for this. Think back to the structure of the cell membrane. Okay, it's a phospholipid bilayer. If you were washing dishes after cooking something that was really greasy or really fatty, if you were to do that just with water, it's not going to be clean. You're still going to see some residue left over. Immediately add in some washing up liquid. That's going to break down the fat, a lot easier to clean with. That's essentially why we're adding in the washing up liquid here. Those phospholipids that we find in the cell membranes, the washing up liquid is going to break them down. So we've got through the cell wall with the knife, we've got through the cell membrane with the washing up liquid, and now we're adding the salt. Because a little bit of the nucleus realistically probably would have been broken up now as well at this point, but just in case we haven't quite got there yet, we're going to add in some salt. And the reason we add in the salt is it's actually going to clump the DNA together, and it's going to make it a lot easier for us to see it in a couple of minutes when we actually get to that point of bringing it out of solution to make it visible. So once we've that done, we're going to add it into a water bath at 60 degrees Celsius for exactly 15 minutes. The reason for this is because the enzymes that we would find with that DNA, it will actually denature them, so the enzymes aren't going to work anymore. We'll then add it into the ice cold water bath. So we'll add it into ice for about 5 minutes. That's going to slow down the action of any enzymes that will remain in there. And then we're going to blend it for about 3 seconds. We're not going to blend it for any more than that because we don't want to destroy the actual DNA that's present in there, but that will destroy any remainings of the cell walls and the membranes. So once you've that done, you're going to be ending up with this solution. It can be quite thick actually, so you have to there can still be kind of some lumps of the fruit and stuff left inside it. So you're going to have to filter it through filter paper in a filter in a funnel and let it down into a beaker or a test tube. So that will collect down here. So we'll have our filtrate. That's going to contain the DNA and the proteins. We actually don't want those proteins in there. We just want the DNA itself. So that's why we're adding in a protease enzyme. That will break down any proteins that are associated with the DNA, and you should just be left with the DNA that's inside here. Now you actually can't see that just yet. So the last step with the ice cold ethanol is going to allow us to actually see the DNA. So DNA is actually insoluble in ethanol. Okay, so if we were to add in, let's say, something like water, we realistically aren't going to see the DNA. But because DNA is insoluble, it doesn't dissolve in ethanol. It's actually going to take it out of solution, so it will make it visible to you. Okay, add in the isocholeethanol, you're going to be able to see that DNA. Now, how you actually add this in, you have to do it very, very specifically. You have to like put it into a dropper, and you have to trickle it down the side of the test tube. I would actually get my students to hold that test tube at an angle as well, and then trickle it down the side, and then very slowly you will start to see the DNA clump together towards the center, and it will appear as this kind of white mass. And then you can use a glass rod if you wish to actually take that out and see it a little bit up close, maybe even examine it under a microscope. But if this was to appear as an experiment question, generally how it comes up is, why did you add washing up liquid? Why did you use a knife? Why did you add salt? Why did you add the enzyme? Why did you add isocholeethanol? So know the reason for each one of the steps, and that's a really handy guide for that as well if you want to screenshot that. So here's an exam question which has come up before based off of DNA. So what do the letters DNA stand for? Well, we know that that's deoxyribonucleic acid. Identify molecule G. So if we have a look at where molecule G is in here, we can see that it's actually complementary to C. C has to be cytosine. So what's complementary to cytosine? That's guanine. The structure label J is a subunit of DNA. Identify the structure label J. So what's the basic unit that's consisting of a phosphate group, the sugar, and a base pair? That's our nucleotide. Name the part labeled K, so K up here. We have two base pairs joined together. They're the complementary bases. What type of bonding occurs? We know that's a hydrogen bond. State one structural difference between DNA and RNA other than the number of strands. So you could say here, DNA has the sugar, and sorry, I have to do this over at the side, sugar deoxyribose. That's not enough to get full marks here. You need to let the examiner know what RNA has. RNA has ribose. Now you will get full marks. You could have also said there that DNA has the base pair thymine, whereas RNA has the base pair uracil. Whatever answer you give there, just make sure you give both sides. DNA has this, RNA has this. And give one non-nuclear location of DNA. A couple of different answers for this. You can say the mitochondria, or you can say the chloroplast. So small amounts of nuclear material found in those structures. Okay, so here's an exam question then based off of the experiment we just seen. Some of the questions you'll see here are actually repetitive from the previous exam question. But how are the two strands of a DNA molecule joined together? That's through hydrogen bonds. Very, very common exam question for that to come up. What is junk DNA? That's DNA that doesn't code for proteins. Okay, so answer the following questions by referring to the procedures that you use to isolate DNA from a plant tissue. Having obtained a plant tissue, example onion, what was the first procedure that you followed? So you cut the onion, you cut whatever. What was the reason for that procedure? It was to break the cell wall. Remember, we have to get through the cell wall, the cell membrane, and the nucleus. Washing up liquid is then used in the isolation. Give a reason for this. So this is to break down the cell membrane. So that will target the lipids in the cell membrane. Salt is also used in the isolation. Give a reason for this. That's to clump the DNA together. What is a protease? So that's an enzyme that acts on proteins. And just a little note there as well, enzymes normally end in ASE. So if you see ASE like catalase, amylase, protease, it's kind of telling you that it's an enzyme. And we see that PROT for protein is at the start of that. So it's letting you know that it's an enzyme that acts on proteins. Not all of them are like that. Another example that would be like that would be lipase. That's an enzyme that acts on on lipids. There's a couple of more other ones as well. So why is a protease necessary when isolating DNA? It's to break down the proteins associated with DNA. So the final stage of isolation involves the use of freezer-cooled ethanol. Describe how it is used. So you just have to say something along the lines of trickle it down the side of test tube slowly. And for what purpose is it used? So it's to bring the DNA out of solution so you can see it. Now, one of our focuses for today is using that DNA structure. We want to see how DNA actually replicates. This is really important to be, for DNA to be replicated when we think of cell division. Our bodies are constantly making new and new cells, and this is all happening through cell division. But for that to happen, we need to have extra DNA there so that it can be passed off to the daughter cells. Because remember, each one of these daughter cells, or each cell that we have, has 46 chromosomes. We can't have any more, we can't have any less. So before the cells actually divide, they need to make sure that they have enough genetic material there to pass off to the daughter cells. So if you think back to cell structure, cell division, which you probably would have looked at the start of fifth year, before the cell can actually divide, it must replicate the DNA within it. This is called DNA replication. It occurs in the nucleus because that's where the majority of the genetic material is found. Okay, and we can see that over here. This is the cell cycle. Cell division is actually a really, really small amount of that cell cycle. Interphase, which is a period where the cell is not actually dividing, can take up to about 90% of the cell cycle. But that DNA replication stage is crucially important to make sure that enough genetic material has been passed off to the daughter cells. Now, how does this actually happen? Before we get into the steps of it, it's just important to know that within the nucleus, within the actual cell, the base pairs that we talked about a couple of minutes ago, there is numerous of those base pairs that just exist there. Okay, they're there in supply and they can be called on whenever they're needed. So there are 4 steps in this replication of DNA. The double helix that we have will start to unwind, and we can see that over here. Okay, it's not the twisted ladder that we see anymore. It's kind of more like just a standard ladder shape. It's just flattened out. But you can see here that the base pairs are still joined together by the hydrogen bonds here in the center. Adenine joined to thymine, guanine joined to cytosine. Now that's actually going to be broke apart. Okay, those hydrogen bonds are going to be broken, and that's going to be done by enzymes. Okay, so the enzymes will break the bond between the base pairs, meaning that all of these base pairs here will actually be exposed. So these existing base pairs that we have within inside the cell, they'll actually enter the nucleus now, because remember the structure of the nucleus, they can enter possibly through the nuclear pores, and they will pair up with the exposed base pairs. Now again, this has to be complementary to it. So this exposed adenine will have to join to a thymine. The exposed cytosine will have to join to a cytosine. And that's the same on the other side as well. This exposed thymine will have to join to an adenine. It has to stay complementary the whole way throughout. So these new base pairs will start to wind up, and they will form a new double helix. So we've gone from having 1 DNA sequence to having 2. But if you have a look, this one and this one is identical to the original one that we had. Okay, DNA replication, crucially important, because this DNA now can be passed off to the 2 daughter cells. Okay, really important for cell division. Now, sometimes this can actually go wrong, and this can lead to a genetic disorder. So a genetic disorder is a genetic problem caused by 1 or more abnormalities that are found in the DNA. This can happen if replication of DNA does not go as planned. So for example, in this sequence over here, in the original sequence, we had a T. Down here, we actually ended up with a C. That's a point mutation because it's happening in 1 point. Okay, so this genetic disorder can happen because of something like that. But we can actually use genetic screening then to test DNA for the presence or absence of genes. So this can be used in adults to test whether they will pass disorders onto their children. And it can also be used in fetal screening to test for disorders before the child is born, so plans can be put into place to make sure that adequate supports are going to be there. Now, slightly different from genetic screening, we're going to be looking at the idea of DNA profiling, coming back to the idea of DNA structure. So DNA profiling, it's a process where a specific DNA pattern called a profile is obtained from a person or a sample of a bodily tissue. Generally, this is going to be used in forensics, so at crime scenes to try and catch a suspect, but it can also be used medically in paternity tests as well. And that's a very, very common exam question to pop up, explaining where DNA profiling is actually used day to day. So how is this actually done? Well, at the crime scene, this sample of DNA was obtained. It was run through a process known as DNA profiling to produce this profile. Okay, the same was done then. They got a sample of DNA from 3 suspects. They created a DNA profile for each one of those. And essentially what we're looking for which suspect has bands that are in the exact same location as the sample that was taken from the crime scene. So we can see here that suspect number 2 has the same bands as the sample from the crime scene. So we can say that suspect number 2 is likely the person that has carried out the crime. Because realistically, you're not going to have 2 people with the same profile unless they're obviously identical twins. Then sometimes you can run into difficulties with that. But it's very, very unlikely, aside from the idea of twins, that you would have the same DNA profile as somebody else. You're going to have a unique DNA profile. It's the same in paternity tests as well. So if we have a look down here, it gets a little bit more complicated with paternity tests because you actually have to look at the bands from the mother and the father. So in this paternity test, this mother here, we have the bands, and we're trying to figure out which of the potential dads is the actual dad to this child. So you have to look at the child and then locate is the band from the mother or the father. So this band here, it's not coming from the mother, but if we run across, we can see it's either going to be dad 2 or dad 3. The band is coming from either one of those. This band here is coming from the mother. If we work our way down, this band here, potentially coming from the mother, but it could have been from this father here. This band here could be dad 1 or could be dad number 3. So dad number 3 here actually has had 2 bands that correspond with the child so far. This band here could be dad 2 or dad 3. So dad 2 at this point has 2 bands lined up. Dad 3 actually has 3 bands lined up. And if we have a look here at this last band, realistically, that's coming from the mother there. So we can pretty definitively say there that dad number 3 is the parent of that child. Now, how does DNA profiling actually happen? So again, like DNA replication, there are 4 steps here. The first one is you need to actually obtain your DNA sample. So you need to extract your DNA. Blood will be an example that could be used for that. Okay, so here's your blood sample. We extract our DNA. That DNA right now is too big to create a profile off of. So we actually have to cut that DNA into segments or into fragments. Okay, enzymes are going to be used to cut that DNA. The fragments are going to be separated based on their size, and I will talk through the process of how that is done in a couple of minutes. It's a process known as gel electrophoresis, but once you've separated all of these fragments based on their size, that's what actually forms all the bands in the profile. You can then compare the patterns. If it's a paternity test between potential fathers and the child, or if it's in forensics, you could look at the crime scene and then potential suspects, okay? So the last step is you're comparing the patterns. So the first one, DNA is extracted. Break down the cells to release their DNA. Very similar to the isolation of DNA experiment. Obviously, it's going to be more high tech than the experiment you would carry out in the lab, but it's a similar idea there. You need to extract your DNA. The DNA is going to be cut, so enzymes are going to be used to cut the DNA into small pieces. Again, even though the enzymes are cutting them, they're not going to be equal sizes, and that's going to be actually hugely important for us in a couple of minutes when we do try to create those profiles. The fragments will then be separated based on their size. So this is done in a process known as gel electrophoresis. So the DNA fragments are going to be placed into a gel, and an electric current is actually going to be passed through the gel. So this is going to pull the DNA from one end to the other. The smaller fragments, they're going to be able to move much faster. They're going to be able to move much further. So they're the ones that will end down towards the end. The bigger ones will remain towards the start. And once you have produced that pattern, you can compare the pattern if it's in the paternity test with the potential fathers, if it's in forensics with the suspects. So there's a very slim chance that 2 people will have identical profiles. So if the patterns from 2 separate samples match, we can pretty much definitively say they're from the same person. So here is an exam question then, based off of what we've seen so far. So the series of images below shows the 4 main stages of DNA profiling technique invented by Sir Alec Jeffreys in 1984. So distinguish between DNA profiling and genetic screening. So we can say for DNA profiling creates a pattern of DNA for comparison, and genetic screening tests for the presence or absence of genes. Number 2, explain each of the stages 1 to 4 in DNA profiling shown above. So step 1 is DNA is extracted. So that was 1. Number 2, DNA is cut using enzymes. Number 3, DNA is separated. So that's based on their size. You could mention there if you wanted that it's done through gel electrophoresis, but it's not actually required in this case. And then you can say there that the patterns are compared. Part 3 we'll do over here. So 2 applications of DNA profiling. So you can say forensics is 1 and paternity tests. And the last question there, identical twins have the same DNA profile. Explain why this is so. Well, they have the same genetic makeup or the same genotype. So that's all you have to say for that. They have the same genetic makeup. Or you can use the word genotype there if you wanted. Okay, so the final topic we're going to look at today then is the idea of protein synthesis, and this is where the RNA is going to be really, really useful for us. So a couple of things just to remember before we start to look at this. RNA stands for ribonucleic acid. Difference between that and DNA, it's single-stranded, not double-stranded, and it contains the sugar ribose instead of deoxyribose. RNA has 3 of the same base pairs. It has adenine, it has guanine, and it has cytosine, but thymine is replaced by uracil. It's single-stranded and it's always going to be complementary to the DNA strand. So the reason this is CUUGUA is because it has to be complementary to whatever the DNA it's matching up with. Okay, DNA and RNA will work together to make proteins. So those proteins we talked about earlier on, where we were talking about the genetic code and the example of the start and stop codons, the junk DNA, all of that, RNA and DNA will work together for that. So for protein synthesis, there are 3 main steps involved. We have initiation, where it starts. We have transcription, where essentially the RNA is copying the DNA. And then we have translation. Now, the first step of protein synthesis, we have initiation. Okay, this is where it starts. So the enzymes are going to start to unwind the DNA. Very like where we seen earlier on with the DNA replication, we move from that twisted ladder shape to just the normal ladder shape. Step number 2 is transcription. Okay, so in the nucleus, that's a question that comes up based off of this as well, is to state the location of both transcription and translation. So transcription occurs in the nucleus. So the RNA bases are going to bond with one of the exposed DNA strands, like we seen in DNA replication earlier on, except instead of existing base pairs joining in there, we have RNA bases are going to join in there instead. Okay, so we can see this up here. This is the idea of transcription. So the exposed base pair here was a T. Down here, it's going to be a U. Okay, we're transcribing it. Okay, if you were transcribing something, you are going to do it word for word the same. So if you're, for example, transcribing something from one language to another, the meaning of what you're doing doesn't actually change. So that's why we have a thymine here and we have a uracil here. It can't be thymine in RNA. It's replaced by a U. The A's are going to stay the same. The C's are going to stay the same. T again will be replaced by a U. That's the only time that will ever happen. A's will stay the same and so on. So DNA is going to act as a template. RNA copies it, it transcribes it, it's the exact same, the meaning can't change. So what will happen then is RNA polymerase will bind the series of RNA bases together to form mRNA. And that mRNA is going to work with another type of RNA, which we will see in a couple of minutes ago, called tRNA, to make the desired proteins. Now, just a couple of things here based off of this. We've mentioned RNA so far. After bringing in this idea of mRNA and messenger RNA, there is 3 types of RNA you're going to come across. You're going to come across mRNA, which is an RNA that's messenger, so it's messenger RNA. We've rRNA, so it's ribosomal RNA. That's actually the RNA that makes up the ribosomes where proteins are actually produced. And we have tRNA, which is referred to as transfer RNA, and you'll see the role of the transfer RNA in a couple of minutes. So what's actually happening in the transcription section here? So we had our original DNA sequence, which we can see up here. That's going to be broken apart. Enzymes will break apart those bonds. The exposed bases are going to be paired up with an RNA. It's going to transcribe it. It's the exact same. The only difference is the T is going to be replaced with a U because we don't have any T's in RNA. And then the RNA polymerase will bind this series together. Okay, so we can see here we have a bunch of triplets or codons. Once we have this messenger RNA, the transfer RNA is going to work to make the proteins, and we'll see that now in a second. So our last step then is going to be translation. So the mRNA will move into the cytoplasm and it will form weak bonds with the rRNA, which is ribosomal RNA. So this is where protein synthesis will occur. Okay. So the tRNA, which is our transfer RNA, this is it down here, is going to carry amino acids to the mRNA with matching codons and attach just after the start codon. So remember, towards the start of this session, we mentioned how we could have junk DNA, which doesn't actually code for any proteins. We have our stop codons, which means your body will just actually stop coding for proteins. And then we have our start ones. So once we hit a start codon, you're going to start to code for proteins. So here is our messenger RNA sequence. Okay, so you can see here you have your base pairs down here in series of 3. So the first one here is actually AAA. So your transfer RNA is going to carry in matching bases. So the matching one for AAA is going to be UUU. Okay, so that's going to attach on here and it's going to deposit its amino acid. So this is actually carrying lysine here. Okay, so this UUU, it's going to attach onto the AAA, it's going to deposit off this lysine, and this tRNA is going to leave empty and it's going to go get another amino acid and eventually bring it back to where there's another AAA. Now what actually happens here, as this tRNA is leaving, it pulls this mRNA chain along, okay, right through the ribosome. So the next one coming in here, this is your other tRNA, it's carrying ASP, it has a CUA to join here to the GAU, it's going to lock on here, it's going to deposit its amino acid, that amino acid will attach onto the previous one to start to form a peptide chain, and it's going to pull the messenger RNA along out of the ribosome. Okay, so the next one will come in here again, it'll deposit its amino acid, push it along, and the next base pairs will move in so that the incoming tRNA can deposit its amino acids and then move along and move along. Once the amino acids are supposed to deposit it off here, this growing peptide chain will eventually form your proteins. We will have to have a considerable number of amino acids there to form a protein, and then will have to be folded into a specific shape. But we can see here how this protein is beginning to form. So your mRNA is going to be coming in here, your tRNA is going to attach onto that, deposit your amino acid, it's going to leave empty and pull the chain with it. Okay, protein will be produced and it will fold into whatever shape is actually required. So here is a question based off all of that we've seen so far. So nucleic acids are composed of subunits called nucleotides. Draw and label the structure of any one nucleotide. Now the reason it's saying there any one nucleotide is because there's actually 4 different options you have there. The reason there's 4 options, you're always going to have your pentose sugar, you're always going to have your phosphate, and then this is going to be your base pair. So for example, I'm going to call that A for adenine. You could have done guanine, cytosine, or thymine. Okay. So the diagram illustrates some of the events. I should just mention on that as well, you would have got 3 marks for each one of those labeled correctly. Part 2, the diagram illustrates some of the events of protein synthesis. What is the name of process K that results in the formation of molecule L? That's going to be transcription. Sorry, that's actually part 1. Part 2, name molecule L. That's going to be our mRNA, or messenger RNA. We were after transcribing that. Part 3, part of the sequence of nitrogenous bases in the gene is as follows. So ACG, TGC, TGA. Using this sequence, write out in order the complementary sequence of bases found in molecule L. So if it was DNA, obviously we would have thymine present here, but it's RNA, so we have to think that we have uracil instead of thymine. So what would be complementary to an A? And DNA will be T, but here it's going to be a U. C would be complementary to a G, G is complementary to a C, T is complementary to an A, we would have a C, a G, an A, a C, and a U. So give the name of the opening through which molecule L leaves organelle J. How does it actually leave the nucleus? Well, we do have these openings on the outside. They're referred to as nuclear pores. Molecule L arrives at structure M and the cytosol is shown. Identify structure M. This is V part 1. This is the ribosome, because ribosome is the site of the protein synthesis, composed of ribosomal RNA. And name the main molecule from which, this is actually the question here, name the main molecule from which structure M is made. This is rRNA, or ribosomal RNA. The abbreviated version is fine there. And describe the main events that occur at structure M which allow for the production of a functioning protein. So you can say that tRNA carries amino acids, to the ribosome. tRNA will bind the mRNA and deposit amino acid. You can say there that tRNA has matching codon to mRNA. You can say that a peptide chain will form. And you can say there that the tRNA pulls the mRNA. And then you can say the protein is formed I'll just have to actually do this a little bit over to the side. I'm running out of space. You can say the protein folds. That would be the easier way of saying it. Protein folds, and that's going to give it its function. Okay, so that was a question based off of protein synthesis. So another really quick one here, kind of just summarizing that. I'm going to fly through this. So name the two purine bases. This is right back to the start. The pyrimidines are the one that have the letter Y. So this has to be adenine and guanine. Describe fully the roles played in protein synthesis by the three different types of rRNA, or three types of RNA. So rRNA forms the ribosome, and mRNA attaches here. You can say that tRNA carries the amino acid to the ribosome and attaches to mRNA, deposits an amino acid. The mRNA has matching codons to tRNA. So you can say that the peptide chain will form the tRNA will pull mRNA through the ribosome. Okay, so once a protein has been synthesized, a final step is required for it to become fully functional. What is this step? And that's the folding. Okay, that's what gives the protein its function. It has to fold. Okay, so we got through a lot of content for today. So just before I finish up, I just want to direct you towards the ExamRevision website where you can access loads of material based on what we've looked at today, as well as for the whole Leaving Cert Biology syllabus. So when you log into the ExamRevision website, sign up, make an account, this is the screen you're going to see. So we've Leaving Cert Biology, we've a bunch of video tutorials, presentations, exam builder, we've quizzes, and we've H1 notes for the entire course. So if we click into video tutorials there, it's split down into the 3 units of the biology syllabus. We were looking today at unit 2, which is the cell. We're going to look at 2.5, and in here you will see that we have DNA and RNA replication and profiling, and we have protein synthesis. There's other topics within this unit as well, so we have genetic inheritance, we have evolution, and so on. But down here towards the end, we can see that we actually have quizzes based on all of them videos as well. So let's say, for example, you wanted to go back over replication and profiling, click into the video, a video will pop up there talking you through everything that we have seen today. Once you've the video done, you can go down to our MCQ quiz. So there's our replication and profiling one. We can see there's 8 questions here. You choose whatever answers you wish. So what's the name of the basic units which make up strands of DNA? That's nucleotides. Submit that. And when you get to the end, it will either go green if it's correct or red if it's incorrect. And you can go back and take it again and again and again until you're fully confident with the material. Now down here towards the bottom as well, we do have a presentation based on everything that you've seen today as well. We do have some notes as well, so you can download these H1 notes covering absolutely everything you need to know about individual topics, okay? There's diagrams and everything throughout each one of those. And right down here at the bottom, we do also have our exam builder feature. So if you click into this, all of the questions based on what we've seen today will appear here. So you can choose whichever ones you want to add to your exam. So let's say, for example, we looked at that one today. Go right back up here to the top. You can download the questions and the corresponding marking schemes as well. It doesn't just have to be the topics we looked at today. If you wanted to throw in some other topics there as well, let's say, for example, you wanted to revise a little bit of ecology as well, you can go back and you can add in some questions based off of that. So do keep an eye on our website throughout the year. Not only do we have Leaving Cert Biology there, we actually have most of the Leaving Cert subjects there. The more subjects you sign up for on a monthly basis, the cheaper it's going to be for you as well. All of our revision courses that happen throughout the year as well will be advertised here. You can sign up for them. So let's go back here to the home screen, our revision courses. You can look back at all of the past revision courses we've done on all of the October midterm breaks, the February midterm breaks, Easter breaks, and then we do some in May in the lead up to Leaving Cert exams. And then any upcoming revision courses will be there in the upcoming booking them as well. So again, the earlier you sign up for those revision courses, the cheaper it will be for you as well. So thanks again for coming to today's Saturday session. There will be lots more biology ones throughout the year, so do keep an eye on our social media channels, keep an eye on our website, and I hope to see you again soon.